Welcome back, folks. Today in this video we're going to tackle more complicated Mendelian patterns of inheritance. In the past we tackled simple Mendelian genetics, where a single character with two alternative forms, two traits, could give you a 3-to-1 ratio of offspring from a mating between two heterozygotes. So big A, small a, mated with big A, small a, would give you a Punnett square with three squares that had the dominant phenotype and one square that had the recessive phenotype. Today we're going to go beyond that. We're going to look at other instances where the molecular chemistry, the biochemistry of the cell, has an influence on what the genes have to say. So where do we begin? Let's look at the agenda for this particular video lecture. I'm going to quickly review the basic patterns of Mendelian inheritance. And then I'll go on to talk about some of these more complicated interactions. In each case, if you understand the molecular biology, you're in a better position to understand the genetics. So the science behind simply means understanding if the cell biology, the molecular biology, the histology, and other forms of interaction can be explained in terms of the genetic interactions. So we're going to look at the science behind dominant alleles, and then we're going to look at the science also behind recessive alleles. I think we'll start with the recessive first because they're easier. Once I've done that, then we'll look at these more complicated interactions, and those represented by the third bullet point all the way down to the bottom. So let's get started. Let's look at a simple Mendelian situation. So this is basic Mendelian inheritance. So in simple Mendelian inheritance, we can either follow a dominant allele or we can follow a recessive allele. It doesn't make any difference. The pattern of migration from one generation to another is exactly the same. So where shall we begin? Let's go and get my color pencil, and we'll start by putting down the parents. So the key in this case is that big A is the dominant condition. So big A gives you the disease, right? Or the condition. Let's just put disease. And of course, the small a, and I wish I had the same type of instruments as Khan Academy, I could type much better. And of course, small a is normal. Okay. There you go, I did my best. That's the word normal. So let's take the parents, and let's take two heterozygotes. So this parent is heterozygous. That's its genotype. And then that parent will be mated with another heterozygote. So there's your second heterozygote. Then the gametes that are produced by each parent will be, in the first case, the sperm will carry big A and small a in equal quantities. So 50% of the sperm are going to be big A, 50% are going to be small a. And then we're going to mate them with the gametes produced from the second parent, the female in this case. So her eggs will carry big A, and another set of eggs will carry small a in roughly 50-50 ratio. Then we need a Punnett square to calculate the probability of the offspring being born. So we take the sperm and we stack them up on top of the Punnett square, and this tells us that we need 2 columns minimum. And then we look at the gametes for the female and we stack them up on the sides of the Punnett square. So that's that big A coming down, and then we bring down the small a. And this tells us that we need 2 rows. So we complete our Punnett square, 2 rows by 2 columns, according to the possibilities with the gametes up here. And then we just bring the gametes together inside the Punnett square. And this will tell us the genotypes of all the potential offspring that can be born to this pair of parents. So there you go. And if you translate this using the key where A is dominant, big A is dominant to small a, then you find that the phenotype here and the phenotype in the box below and the phenotype in the box to the right, all three phenotypes are the same. So we get 3 boxes out of 4 being of the dominant phenotype, and we just get the 1 box at the bottom of the recessive phenotype. We get a 3-to-1 ratio with respect to the phenotypes of the offspring. If we now perform a similar type of genetic cross, this time with a recessive condition, where the small a is the diseased allele and the big A is normal. So big A allele equals normal, and the small a would be, in this case, afflicted, i.e. diseased. Okay. And while I'm doing this, you can most probably predict ahead. So again, we're going to start with the heterozygous parents. So big A, small a, multiplied by big A, small a. And you'll see that when you get to the gametes, we have big A sperm and small a sperm. We have big A eggs and small a eggs. And then when we put the Punnett square together, the Punnett square will look exactly like that. And this time, we also get a 3-to-1 ratio of the phenotypes. But this time, the diseased condition is going to be present only 1/4 of the time compared to the normal condition, whereas over here, the disease condition was present 75% of the time compared to the normal condition. As you already know, recessive alleles are much more common than dominant alleles, and we'll discuss that in a few seconds. Let's continue this analysis and look at sex-linked dominant and sex-linked recessive alleles. Because the X-linked condition is much more common, that's the one we're going to consider. Because the X chromosome has much more in terms of genes than the Y chromosome in humans, so it's more likely that the inheritance will have to do with the X chromosome. So we're going to keep the same terminology as up here. We're going to use parents and gametes and Punnett squares. So let's get that going. Let's put the key together. So in this case, let's use our symbology. So we'll say if it's dominant, let's give it a letter that corresponds to dominant. Okay, let's use big D to be the disease condition, right? And then we'll use a small d to denote the normal wild type condition. And if you want to put a plus next to it, you can, and the plus simply signifies that that allele is wild type and normal. So where do we go next? We should move on to the parents and type in the phenotype of the parents. So we're going to mate. In this case, we're going to put the male first and the female second to keep things consistent. And we're going to mate a male that has the disease with a female that doesn't have the disease, right? So a male has a disease, so the phenotype of that's the phenotype of the male. So the corresponding genotype of the male, because the male has one X and one Y, we're going to have him have the disease, so big D. Okay? And then we're going to mate him with a female who has 2 X chromosomes, and she is going to be normal. So D, small d, small d. And then we do the gametes, and from the gametes we can see that he produces 2 types of gametes, those that contain the X chromosome and those that contain the Y chromosome. So in this case, the Punnett square will have big D above one column, and the Y chromosome will be alone in the other column. And she will produce just one type of egg. So all her gametes will contain the X chromosome with the normal small d. So we just need a Punnett square that corresponds to that condition. So there you go. So this time the Punnett square doesn't have to be double stacked, i.e. 2 rows, it can be a single row, but it needs to be 2 columns to accommodate the difference in the gametes in the male. So bring the Y down from the sperm and carry over the X from the egg and maintain the allele. So that's a D, small d. And do likewise with the sperm that carry the X chromosome. And in this case, they carry the big D, the diseased version, and then the egg delivers the normal type of small chromosome. And that's it. So all the offspring for this couple here, or this mating, will be this particular genotype, right? So the ratio will be one of these to every one of those. The phenotype will depend now on our key. In this case, all the females will have the disease, and all the males will be normal. Okay? And we can indicate that by using the international symbols for males and females. There you go. Oops. That should be a like that. So we've done that. Now, with sex-linked dominant alleles, you've now broken this 3-to-1 ratio that Mendel had come up with. So this is an indication, this is a departure, this 1-to-1 ratio in terms of their sex is an indication that this condition is sex-linked. Okay? If we do the reciprocal cross, where the male is now normal and the female is diseased, then let's see what happens. Do we maintain this 1-to-1 ratio, or do things change? So let's figure it out. So the parents in this case are going to be, again, a male, put the chromosomes down, and a female. And now we're going to designate the different chromosomes with the true designation. So the male is going to be normal, so we'll give him small d. The female will be diseased. Now she can be diseased in two different ways. She could be a homozygous, diseased dominant condition, or she could be a carrier. So she carries the normal allele, and she's got the disease on the other allele. Now the term carrier is not normally applied to the dominant condition. Maybe we should use the word carrier on the other last box when we get to that. So in this case, we just consider her as being heterozygous. Right? So she's going to carry a small d on this X chromosome and a big D on the X chromosome. So when it comes to gametes, he will consistently produce two types of sperm, X small d and Y. So we can put that on top of our Punnett square. So this is one type of sperm. Then over here we have the other type of sperm, right, that he produces. In her case now, we do have two alternative eggs. Those eggs that carry big D and those eggs that carry small d. So let's put that here. So let's take this chromosome and transplant it here. And then we'll take the bottom chromosome to be the one that carries the big D. Now we have a Punnett square. We put that Punnett square together. And you'll see it's quite straightforward to put the Y straight in, because we know they're going to be there, right? And then bring over the chromosomes from the egg. So that's going to be small d, that's going to be large D. And likewise, we drop down the chromosome from the sperm, small d, X, and then we bring over the chromosomes from the egg, big D there and small d here. You can already see that the genotypes are variable, and the corresponding phenotypes, let's work them out. So the males, which are this column, they contain one box that's got the normal condition and another box that has the disease. So in the males, there's a 1-to-1 ratio. And in the females, we have homozygous normal, and we have heterozygous, and that would be the disease, right? So in both cases, in both sex, we have a 1-to-1 ratio between the disease and the normal condition. Unlike this, where the males were one particular phenotype and the females were the other, in the second example, there's no such differentiation, because we'll have two types of condition in each of the females and the males. So that's what happens in that case. Over here, let's just go and finish off by saying that the big X there carries the big X. That means the first X carries the normal allele, the wild type, and the second one carries the disease. And what would happen here? Well, nothing really, except that you need two small d's to be normal and heterozygous and homozygous dominant. For the big plus sign would be making you, in this case, normal. Okay, so let's just put one cross quickly together. Again, we're going to start with heterozygous parents. So we're going to have the male and the female. And as I keep saying, practice makes perfect in genetics. So I've taken so long to illustrate this, but this is a review slide of previous material. So many of you may be familiar with this. So what do we start with? Let's start with a male that's normal. So let's put the plus sign here. And let's start with a female that's got the disease. So must be homozygous recessive, right? And then we calculate the sperm. So this sperm will be normal. That gets that chromosome. And the other sperm will be carrying the Y chromosome alone. And then in this case again, because the two chromosomes are the same, they carry small d, we just need one representation for all the eggs. Then we put this Punnett square together, and then we'll end up with something of this particular genotype. There you go. And then we'll have the same disease chromosome coming in from here, and then we'll copy this chromosome down and put it here. So this column represents the females, and this cell, I should say, corresponds to the males. The females are all normal, and the males have the disease. Very similar to this. And then, just to prove that we can do this, we can do the reciprocal cross, right? And in the reciprocal cross, we'll have small d over here in the father, and then we'll mate him with the double wild type homozygous dominant female. And we'll find that her chromosomes, her eggs, those chromosomes contain just the wild type condition. And in the father, we have the two types of gametes. So these are the gametes, and that is the egg. And we bring them together just like this. And we find that the males are all going to be wild type, and the females are all going to be wild type, but they're going to be carriers. So a carrier is an individual that is phenotypically wild type, but one of the alleles is for the recessive diseased condition. And here, the ratio between the two would be males and females, 50-50, but all the individuals would be normal. You can try that yourself by making the mother, the female, heterozygous, and then see what happens. You'll end up with another column, and you'll have to deal with that in due course. Before we move on to look at the biochemistry, it's important for you to know that analogies are very important in understanding molecular processes. One thing that you may have learnt in a previous course is that a cell can be considered like a city. So the nucleus of a eukaryotic cell could be the downtown area, where the offices of the council are, where decisions are made on how to run the city. And then the cytoplasm can be where the community of people resides, where the factories are, and where the infrastructure of the factory, where the infrastructure of the city takes place. So if that analogy is now extended to understanding the concepts in this chapter, that may make it easier for you to be able to relate to some of these more complicated interactions that take place inside cells. So let's try that. Let's try this understanding. So let me get my marker here. And we're going to outline this particular city. So the red is a city. And inside this city, we have a factory that is producing goods. Okay? So here's our factory, and I'll only draw it once because it takes time. So the factory is akin to the DNA and the nucleus. So within the factory, instructions are provided to manufacture goods. And those goods are produced inside this factory. And then they're exported. And once they're exported, they take a journey to their destination. And the destination may be your home. So you ordered something and it's arriving from somewhere, right? And the journey itself takes place on a road like this. And on the road, there may be a delivery vehicle, like a UPS or something, or a truck, right? So let's put that van in there like that. There you go. Not very good, but good enough for our analogy, okay? So instructions for making stuff are in the factory. The factory has the raw material to produce the goods. The goods are loaded onto the transport vehicle. That takes a journey across the distance from the factory to your home, and then it gets to your home and it makes a delivery, and then that delivery is available for you to unpack it, to unwrap, and then to deploy, right? So this analogy works. So let's put that in the context of a cell. Let's change the color, and let's choose blue to represent a cell. So here's the cell, and we're going to stick with the eukaryotic cells. But prokaryotic cells do things in a very similar fashion. And then we have a nucleus. And inside the nucleus, we have a pair of chromosomes, homologous chromosomes. So this is a diploid cell from a eukaryote. Let's assume that on the surface of that DNA, we have two genes. One gene is right here, and the counterpart on the other chromosome is right there. And in this case, the two alleles are different. So let's make them different. So let's call one of them big A and the other one small a. So we have a heterozygous condition. Okay. So how does that work with respect to the analogy here? Suppose the product of that gene is required in the cytoplasm over here. So this part of the cytoplasm requires the products of those genes to be utilized here. So we put your house over here and we'll say that we need the products here for the cell to function normally. So what happens is this. The genes are turned on, they are expressed, and then they are transcribed into RNA. Now, what happens? Well, in some cases, this gene is broken. It doesn't have enough information, maybe at the beginning of the gene, maybe in the promoter. So the gene is broken. Let's assume it's completely broken. So this conveyor belt in the factory is broken, so it produces nothing. And this one is working normally, so it produces RNA, and then the RNA is going to make its way to the ribosome, which is another factory that sits here inside the cytoplasm, and this produces a protein. Let's represent the protein as a green blob. Then the protein has to be delivered to its destination, and once it arrives at the destination, that protein can do its particular job. Now the cell is happy, everything is fine. Now the question is, when only one conveyor belt is running in the manufacturing plant, is that enough to satisfy the needs of this part of the cell? In the cases that we considered in previous examples with Mendelian genetics, the answer would be yes. One allele is enough to satisfy the needs of the cell. Therefore, if the other allele is broken, then there's no effect on the cell. It behaves the same as if you had two big A's and you were producing twice as much of this green blob, the protein. So recessive alleles can be explained as broken alleles, where the final product is not made or delivered to its destination. As far as the cell is concerned, it's looking right here. Do I have any green blobs? And if the answer is no, then the cell will exhibit a diseased phenotype. But if it has enough from a heterozygous condition, then everything is all happy. So just to make sure you understand this, what would happen if the second allele here was a small a? If you had time to think about it, then the analogy will be that neither conveyor belt is functional. There's no product, so there's nothing to deliver to any destination. Therefore, the final destination gets nothing, and that can manifest itself as a defect in the cell. So, pretty easy to explain at this particular juncture. Let's move on to the next slide and look at how dominant alleles work. With dominant alleles, the situation is a bit more complex because we've discovered through experimentation that the way that dominant alleles work can fall into three main categories. So let's look at each one very quickly. So let's reconstitute our cell. Here's our cell, here's the nucleus, and within the nucleus we have a pair of homologous chromosomes. And on those chromosomes we have the genes that we're interested in. So what we're going to do now is demonstrate how a dominant allele may have a functional effect on the cell. So let's pick another color. Let's go to blue, and we'll use the blue to demonstrate things. So this is big A, and this is small a. So now we have a heterozygous condition. Remember, in this case, the small a is the normal wild type allele. Okay, so this can be wild type. So what happens? So the small a works normally in that it produces a messenger RNA. The messenger RNA goes to the ribosome, and then it gets translated into a protein, and then the protein is delivered to a destination inside the cell. X marks the spot. Okay? So how do we explain a detrimental effect on the cell from the dominant allele? And the answer is this. In one case, the dominant allele expresses itself much more readily than the normal allele. So this produces probably, maybe in one case, more RNA. The additional RNA produces a lot more of the protein, and then the destination gets overwhelmed by the presence of the protein, which could interfere with some biochemical process, or it could crowd out something else by being present in too much. So this is the example of too much of something can be a bad thing, right? So that's a gain of function. So the mutant allele can be overexpressed, or it can produce a lot of protein, and then that protein clogs up the cell internally. So let's move on to the second example, dominant negative. So the word negative means that whatever is produced from the dominant allele may have a negative interaction within the cytoplasm. And indeed, that's the case. So let's go back to our original condition. Let's get rid of all the proteins and the RNAs. And then we're left with, I got rid of the cell too. So let's put the cell back. I'll keep it in a bluer color, but this time it's a light blue. So it doesn't matter, we get the point. So let's change the pen color to red. Purple, let's try purple now. So what this means in this scenario is that the wild type allele, the small a, produces messenger RNA. The messenger RNA gets coded and translated into protein, and the protein is produced, and then the protein is delivered to the destination. In this second example, the expression of the big A allele, the mutant allele, produces a messenger RNA. The messenger RNA also gets translated into a protein, but that protein has an altered configuration, an altered shape. It doesn't look like the wild type blob. It's no longer circular. It's more triangular. And when that arrives at the destination, maybe it has a negative effect on the system to which it's designed to complement. So wherever it's needed inside the cell, it doesn't work. In fact, not only does it not work, it gets in the way of the normal protein by hindering its ability to do its job, like interference. So that will have a negative effect on the cell. So we call that dominant negative, because if the big A wasn't there, the cell would work fine. But because there's one copy of big A allele, it's enough to upset the cell, and therefore big A works in a dominant fashion. Let's turn our attention to the third mechanism that we've uncovered, haploinsufficiency. If you look at all three terms, they give you a clue as to what could be happening at the molecular level by using simple English. And haploinsufficiency is no different. Haplo means a single copy. Diploid means two copies. And insufficiency means something is not enough. So maybe in haploinsufficiency, you need two copies of the alleles to do a good job. One copy will not be enough. And that's exactly what happens. So let's go back and explain this. So let's get rid of what we don't need. Let's get rid of these triangular proteins. Let's get rid of the RNA. And let's go back to this exact same situation. So what it's going to entail is that the A allele, the big dominant allele, is broken. So it doesn't produce RNA and it doesn't produce protein. But the recessive allele, the wild type, produces RNA and it produces protein, the same globular protein that we were looking at earlier. But this protein, when it arrives at its destination, there's not enough of it to help with the cell's functionality. The cell needs 2 doses, but it only delivers 1 dose. So there's a deficiency in the cell. Haploinsufficiency, haplodeficiency. So think about that. So go back to the example of the factory. Suppose there are 2 conveyor belts producing products to satisfy demand, toilet paper maybe, and that one of the conveyor belts has broken down. Now, even though one conveyor belt is producing toilet paper, that's not enough to satisfy the needs of the community. Therefore, there's a deficiency. Let's move on to this concept of penetrance. Again, if you look in the English dictionary, the word penetrate, from which penetrance comes, is to probe deeper into something. So when you penetrate something, you enter something, or you go deeper into that environment. So penetrance can be used in the concept of a cell to explain how there could be a defect even though the gene is normal. I brought back this figure here because it would make sense to explain it in that context. Suppose we have a factory, the nucleus producing product, and the product is normal, and then it's loaded onto the truck, and the truck never makes it to the destination. So as far as the cell is concerned, it's a defect because it doesn't receive the commodity, and therefore the city suffers. There's nothing wrong with the DNA. There's nothing wrong with the allele, but there's something wrong with the mechanism of transporting it to its final destination. So that's important in genetics. And if you look at this, this would make perfect sense. You order something from the factory, they make it, they put it on a truck, the truck breaks down, you'd never receive the product. As far as you're concerned, you never got something. To explain this further, I brought back the image of a cell. Let's assume that the product of the dominant allele is necessary to produce a defective phenotype inside the cell. So the allele is there, you have the allele, and you should be expecting to get the condition because it's a dominant condition. So let's follow what could go wrong in the same context as our factory example. So let's use a different color and we'll illustrate that with the orange. So the gene is there, is working normally, and it produces an RNA. The RNA connects to the ribosome and it forms a mutant protein. And that should change the biology of the cell to become mutant. But because of the environment or something like that, the protein never makes it to the destination. If the protein doesn't make it to the destination, it doesn't matter if you have the allele. You are not producing the end result. Therefore, this cell would be normal if the small allele is working and it supplies everything it needs. So how would that work? Well, the small allele produces messenger RNA. That messenger RNA produces a normal protein, and that protein, for some reason, gets delivered. Therefore, the cell would behave as though A was normal, a small a, and it wouldn't have the disease. Because people live in different environments, maybe this individual lives in an area where the mechanism does deliver the mutant protein. And another human being maybe lives in an environment where inside their cells, the mutant protein is not delivered. So the environmental conditions can influence the delivery or no delivery of that product. As far as the individual is concerned, they're either going to get the product or they're not going to get the product. So they either have the effects of the mutant allele or they don't. But in the population, you can imagine 50 people have the effects of the big A and maybe 100 people don't. So in that case, the penetrance of the allele can be considered to be 33%, because 33% have the defect and 66% don't. So penetrance can have a population value, but at the individual level is all or nothing. Either you have or you don't have. So if somebody gives you a question on penetrance that has a percentage, they are dealing with the population of organisms, not an individual. Keep that in mind, please. Before we leave this particular topic, you may be thinking between the lines, hang on a second, maybe in a particular environmental circumstance, maybe some of that protein is delivered in my cells. Not all of it, not none of it, but some of it. Would that not affect the penetrance? Indeed it would, and we have a term to cater for that particular situation. The expression of the allele. The allele is there in order to have its effect. It has to be expressed all the way down to the level of where it's needed by the cell. So expressivity can have a value. So if you have, as in the classic example, the dominant gene or allele for multiple fingers, polydactyly, then in some people in the population with that particular condition, they may have a tiny indication that they have an extra finger. So the expressivity of that allele is very low. You can have another person that has 2 fingers on each hand and each foot. Therefore, the expressivity of the same allele will be very high. So the analogy to go back to the delivery track is that in different circumstances, in different people, the other genes and the environment may allow the allele to have a major influence all the way down to no influence on the phenotype of that individual. This brings us naturally to another very important concept in biology and genetics. And this concept says that whenever we consider characteristics, we don't find anything that's 100% genetic all the time. And we don't find anything that's non-genetic at any time either. So the 2 forces, the DNA that your cells carry and the environment in which those cells exist, the 2 forces are interacting constantly. So the range can go from 5% genetic and 95% environmental. So the environment has a large influence on dictating the outcome for that individual, all the way to the other side where you could have a 5% environmental and a 95% genetic interaction at the chemical level. And genes can be anywhere in between. For instance, there could be a certain type of lung cancer that's very highly environmental, but not 100%, because there's some genetic influence on it too. Or you could have a certain type of cancer where it's genetic and you inherited the cancer from your parents, certain types of colon cancer, where the environment has a small role to play, but it's going to be mostly genetic. So if you eat healthy food and you eat plant-based foods, maybe those individuals will not develop any cancer, but their brothers or sisters that have a bad diet, fatty foods and processed foods, maybe they'll develop cancer. So the environment is always there, operating at some level with every gene. Other examples that are used to illustrate this concept would be the type of animal that lives in the Arctic fox. So during the winter, when the environment is white because of snow and ice, it makes sense for the fox to be camouflaged. So the entire fox has white fur. In the summer, when the ice has melted and the environment has become more brown, the fox changes color to a brown coat color, and therefore it can camouflage in that particular environment. So the environment influences the behavior of the fox's genes. The other great example is medical intervention. So if a baby has a phenylketonuria, PKU, then if the diet is not modified, it will develop some cognitive issues, brain damage, and other developmental problems. However, if it's detected very early on within the first few days of birth, then we can change the diet to avoid a certain amino acid, and the outcome for the baby is as near normal as possible. Since the environment has a say in the outcome, we have to then be able to quantify the environmental component. One way to do that is to take identically cloned animals, genetically identical, with the same genotype, and then experiment with them at different environmental conditions, and then figure out which allele gives the individual in the population a particular phenotype. And that way you can decide on the phenotypic effects based on the environment. And that is called the norm of reaction. So for a big A allele, you can decide what the norm of reaction is under a particular set of conditions, and then compare that with a heterozygous condition or an even homozygous recessive. And that'll give you a profile. A better way to look at it is the range of conditions under which that particular genotype is able to stabilize and produce a certain phenotype. Beyond that, a different phenotype may be generated. The next three alterations in the dominance perspective that we're used to seeing, where there's complete dominance, are these three: incomplete dominance, overdominance, and codominance. Each one is easily illustrated with an example. The example that we use for incomplete dominance in almost every genetic situation is the 4 o'clock flower. Let's illustrate that with a nice example. What we're going to do, we're going to take a pure breeding red flowered plant and mate it to a pure breeding white flowered plant. So in terms of genetics, each plant is pure. Therefore, it doesn't have a heterozygous condition. So let's say that the parent on the left-hand side is red flowered. So that's its phenotype. And its genotype would then be something like this. So C stands for the color gene, and this is the allele that we have. Big R stands for red, and small r will stand for white. So because these plants are diploid, we need 2 chromosomes in each individual. So that's one parent, and then the white parent will be the following genotype. So that's going to be small r. And because it's pure breeding, both chromosomes will carry the same allele. Then the gametes that these parents will produce will be C big R for the pollen, and C small r for the ovules inside the female parts. And the offspring would be heterozygous. So the offspring would be big R and small r. And their phenotype we would expect to be red, because that's what we're used to. But in this case, all the offspring are pink. And the answer, well, not the answer, the question is why? And we can explain that quite nicely at the biochemical level. So let's look at an example of a cell. Here's a cell again, magically appearing. So let's pull that up here, and let's get rid of what we don't need for now. So I'll get rid of the RNA and the DNA. So I have to do some magic to that first in order to do exactly what I want to do. And let's get rid of this, and this, and that, and that. Okay, so we're looking at this particular genotype. So plants have a nucleus, and inside the nucleus they will have 2 chromosomes, just like you and I. And on the chromosomes, in this particular child, F1, we have big R on one chromosome, and then we have the small r allele on the other. So what's going on? Let's assume that the small r allele is broken. It can't produce any product. Therefore, it can't contribute to the coloration of the petals. So this particular C gene is responsible for making pigment in the petals of the plant, the cells in the petals. In this heterozygous condition, we're going to compare that to the homozygous condition. So using a different color, I'll go back and illustrate outside what the DNA of the homozygous parents would look like. So this one here will be this plant. Let's put the DNA of that one together so we can see what that would look like. We'll use the same colors as over here. So we have an allele on this one and an allele on this one. So in this case, we have 2 broken alleles, small r and small r. Therefore, neither one is producing any messenger RNA. Neither one is then producing any pigment, the protein pigment, and then the petals will not be colored. And the petals will be the default color, which is their background color. And in this case, because nothing is being delivered to the petals will be white. So that should make biochemical sense. In the case of the parent over here, the 2 chromosomes will be indicated right here, and both chromosomes are working as expected. They're both producing messenger RNA, and from that messenger RNA we have pigment produced. So this allele is producing pigment at 100% of its own capacity, and then this allele is producing pigment at 100% of its own capacity, and that pigment is then delivered to the petal, where the petal then has a red phenotype. So to get red, you must have both alleles working as expected. So what happens in the children to get the pink phenotype? Well, only one of the chromosomes, the big R, is functional, producing RNA, and then that produces pigment, and that pigment is then delivered to the petal. But the other chromosome is broken, and there's a block, and there's no pigment produced from that. So the delivery of pigment is 100% from the first chromosome and 0% from the other chromosome. But that's not enough to generate the full red color, so you get a dilution effect, and the petals are pink. So basically they receive only half the pigment that they need to become red. So pink is the outcome. So this is an example of incomplete dominance, where the heterozygote is in between both homozygous individuals, right? Homozygotes. So that's easy to understand at the biochemical level, more so than maybe at the genetic level. And just to finish this analysis, if we were to take this individual and cross-fertilize it with another individual of the same type, or self-fertilize it, either way you get the same result. And the other individual is heterozygous too, what do we predict? Well, the gametes from this will be big R, small r, and the gametes from this will be big R, small r. So let's put them together. So big R from the parent on the left and small r, and they go above the Punnett square. And then you have small r and big R gametes coming from the second parent on the right. And that gives us a Punnett square that looks like a 2 by 2. And we complete that like this. Here we go. And then we have big R and big R coming into these offspring, the F2. And then we have big R, small r coming into these offspring. And we have big R, small r coming into these offspring. And then here we have small r, small r. So when we did this with the typical dominance recessive relationship in Mendelian genetics, we got a 3-to-1 ratio in terms of the phenotype, right? So let's have a look at this. So this box here says that all the offspring of that type will be red. Then this box here says that all the offspring will be white. And then these two boxes, they give us the heterozygote. That will be pink. So instead of getting a 3-to-1 ratio, what we do get is 1 red to every 2 pink to every 1 white. So we get a 1 to 2-to-1 ratio. And that is normally indicative of incomplete dominance. Next, we move on to the middle term, overdominance. Now, overdominance has an alternative name, and you may know that as heterozygous advantage. In this particular case, the phenotype of the heterozygote is going to lead to more offspring. So from the natural selection perspective, it's advantageous to be a heterozygote. So let me give you the example of sickle cell anemia. It's classically used to explain this. So sickle cell anemia is basically a disease, a recessive disease of the blood. The red blood cells, which contain hemoglobin, that hemoglobin is a protein that forms a structure that can carry oxygen and carbon dioxide. Under these conditions in these individuals with the disease, the hemoglobin molecules, they get connected in a long rod, and that rod can change the shape of the red blood cell into different shapes. And under certain physiological conditions, when there's low levels of oxygen, when the individual is stressed, physiologically stressed, these alternative shapes can manifest inside the red blood cells. And instead of the red blood cells being concave shaped, which is this shape, which is designed for their function, they actually become sickle shaped like that. And the sickle shape is not conducive to traveling through capillaries. And they form obstacles like dams in the blood vessels of these individuals. And they deprive the tissue downstream of oxygen. And that can cause pain and tissue damage and tissue death. The long-term outcome for these individuals, homozygous recessive, is a reduced life expectancy, unfortunately. The heterozygote and the homozygous dominant, they have a normal life expectancy. So how does this work? Let's have a look at the genetic makeup, and then we'll look at the environment in which these individuals benefit. So where do we start? So we like to use the classic examples of alleles. So HB stands for hemoglobin. And what we're going to do, we're going to use two examples. We're going to say HB plus is the wild type. That makes the normal type of hemoglobin. That leads to the biconcave shape of the red blood cells. And then we have the alternative diseased allele, which we call HB S, small s, right? And that'll stand for sickle cell. So there's our key. And let's have a look at what happens. So with these two alleles, there's three possibilities for a diploid organism. I'll just use the plus sign and the S sign to save time. So the first possibility is that you could be an individual who is, or she is, homozygous dominant. So this person does not have sickle cell disease. Then we have an alternative example of a person who's going to be heterozygous. And they are called carriers. They have sickle cell carrier, heterozygous. And then we have the third example. And these people are homozygous recessive. And they are the ones who have sickle cell disease. Sickle cell trait is also a name that's used for this. So how does this work? Well, let's look at the conditions in which this works. So let's look at transporting blood, right? So let's look at hemoglobin and blood. So the blood of these individuals is absolutely normal. The blood of these individuals is also normal under normal circumstances, but under certain circumstances it can sickle. So it's like halfway. And the blood of these people is sickling most of the time, causing the early death. So in this particular example, it's best to be dominant, homozygous dominant. But in parts of the world where there's malaria, the malaria parasite, Plasmodium, does survive extremely well in these individuals, but it doesn't survive well in the heterozygote, because as soon as the parasite enters one of these normal red blood cells, it sickles. And once it sickles, the mechanical shape change and the physiological changes, they also kill the parasite. So it's really, so that should be an X. It's really advantageous to be heterozygote in those parts of the world. And what about these people down here? Well, they also have an advantage from having their red blood cells sickle. But of the three categories, the best is the middle one, where you have an advantage against the malaria parasite and your blood doesn't sickle. So this is called the heterozygous advantage, or overdominance. There are additional examples of overdominance that can be explained at the biochemical level as well. So for instance, let's assume that we have three phenotypes. Let's type them in. We'll just call them A for now, just for the sake of simplicity, right? So we have the homozygous dominant, we have the heterozygous, and we have the homozygous recessive. And in this case, what is it best to be? Well, let's have a look. If the A allele produces an enzyme that works at 37 degrees Celsius inside the human body, and the small A allele produces an enzyme because of its genetic alterations, say it works at 33 degrees Celsius, best. So individuals that are homozygous dominant, they will produce from each chromosome a copy of their protein that works at 37 degrees. So 37 degrees from that chromosome and 37 degrees from that chromosome. So under normal conditions, those people have a nice advantage. The homozygous recessive, that will produce from its chromosomes a version of the enzyme that works at 33 degrees. And likewise from the homologous chromosome. The heterozygous, on the other hand, will produce enzymes of both types. One that works at 37 degrees and another enzyme that works at 33 degrees. So from an environmental perspective, these people do best at 37 degrees. These people do best at 33 degrees, but the heterozygote, it does best at a range of temperatures from 37 degrees down to 33 degrees. So which one of these would you like to be? And the answer is, it may be advantageous in a cold climate to be this particular phenotype. So another example of heterozygous advantage. So let's move on to the last example of codominance. Co means together. That means both of them, both alleles, have exactly the same power and they both exert themselves at the same level. And the classic example of that is the human blood group, the ABO blood group. So let's try to quickly explain that. So unfortunately, we have 3 alleles this time. So this may be the first time that you have come across a gene that has alternative alleles. In fact, most genes have lots of alleles. There are only a few genes that are like Mendel's examples, with big A and small a. Most of our genes have multiple alleles. So we're more sophisticated than we may look. So in this particular case, we're going to use a certain terminology where we use the I to represent this particular gene. And the gene comes in 2 flavors. That's going to be blood type A and blood type B. But there's also a 3rd allele that exists at this gene locus, and that's called I. Now, I refers to blood type O, and I represents a broken system. I is recessive to the other 2. So we have an example of recessive and codominance all in the same system, right? What does that mean? Well, let's look at it at both the genetic level and the biochemical level. So let's put the genetic level here and the biochemical level here. And that will make complete sense, hopefully. So let's go for various combinations of these 3 alleles, right? As you know, when you have 2 possibilities and you have 3 alleles, that will lead to 8 different types of phenotype. So you could have somebody who has big A, big A. And what this does is that the big A codes for an enzyme, and that enzyme sticks on the surface of your blood cells a particular sugar. And that sugar is recognized by the immune system to be type A. Therefore, the immune system is educated not to attack type A because the cells have that on their surface. So in this case, the surface of the red blood cells, so here's a red blood cell, and it will be expressing A and in its nucleus, capital A, in both cases, and this will then be deposited on the surface of the cell as an A-type antigen, right, on the top of these sugars that are coating the outside surface of the membrane of these red blood cells. Because both copies of the gene are the same, both alleles are the same, they both contribute to the delivery and the expression of A. So that's what happens there. So likewise, very quickly, a person who's homozygous B, they will express on their red blood cells just the B antigen. The B antigen is chemically different. It has a different shape and it uses a slightly different chemistry, which is noticeably different from the A. So the immune system of this individual recognizes that and it then protects the body from attacking the B antigen. And then, of course, we have the I, homozygous I. So these people are going to be blood type O. So let's put the blood type O in between. So this is going to be blood type A. Okay. So they have 2 chromosomes, and both chromosomes are defective at that particular gene locus. So neither one is producing any enzyme. So the surface of these cells is missing the A type and the B type. It doesn't have any of these particular antigens. So it's not educating the immune system to be sensitive to either one. But we can have other cases too, as I said earlier, right? So just put a couple down. You could have A, B, where this individual inherited A from one parent and B from the other. So what would happen in this case at the biochemical level? Each chromosome, the A and the B, would be expressing itself, and this will deliver A to the surface, and this will deliver B to the surface. So this cell will be covered in A's and B's. So this will be blood type AB. What other alternatives do we have? Well, we could have this with A and I. So once again, we have a condition, heterozygous, where this is producing A and delivering A to the surface, and the second chromosome, because it carries the recessive version, is broken. Therefore, this particular individual will only have A antigen on the surface and nothing else. And this person would be blood type A at the phenotypic level. And we can keep going on this analysis, right? So you could have B with I. Regardless, what we have here is a nice demonstration of the relationship between these two, the A allele and the B. So when they're both present in the heterozygous condition, the individual has codominance. Both are expressed equally. Just to finish off this, the blood types are very important. So if you're blood type AB, your immune system is educated to receive blood from anyone. You could receive blood from an O-type donor. Because their cells are not covered in any antigen, there's nothing to attack, so that blood will be accepted by you. How about blood type A? You could accept blood type A because your immune system has already been educated not to attack A because you carry A on the surface of your cells. So you could accept that. How about B? You can accept blood type B as well because, likewise, your immune system knows about B and it doesn't attack B. And of course, you can accept blood from somebody same as you, AB. So a person who's AB is said to be a universal blood acceptor. They can accept blood from any other blood group. Let's have a look at blood type O. Blood type O can only accept blood from an individual that's blood type O. If you give this person blood from a blood type A, then those blood transfusions coming into the body will carry A on the surface, but this person's immune system has never seen A ever before. So it'll treat A as a foreign infection and it'll start attacking those blood cells. And that can lead to some detrimental outcomes for this patient, including death. So A is no good. Likewise with B. If you put B into this patient, who's blood type O, then that's not going to be any good either. And if you give them AB, then you get a double whammy because the immune system will attack both the A and the B on the surface of the donated blood. So that's no good for either. So O can only accept from another O, and you have to be really careful if you're blood type O. But O is a universal donor. So if you're blood type O, you can give your blood to anybody else because your blood contains nothing on its surface and it doesn't cause a reaction in anybody else, right? Blood type AB is a universal receiver. They can accept blood from anybody else. What about if you're blood type A? Well, you can only accept blood from an O or another A because of the same reasons that we gave you there. But you cannot accept blood from a B, type B blood donor, right? Likewise for blood type B. O is okay, B is okay, but A is forbidden. And of course, AB would be forbidden too, because that contains some A in this case. And over here, for this individual, it contains some B, and that would be not good either. So there's a good analysis of codominance. But this system is mixed because I is recessive to both A and B. Let's move on to the next topic, which is sex influenced. So again, the English language helps us. It means the sex of the individual will influence the outcome, the phenotypic outcome of these particular alleles. So let's look at that very quickly. So say we have a male over here and there's a female over here. So we expect there to be a difference in the outcome for the same gene that's 6. sex influence, right? So let's go for the homozygous condition. And let's go for the heterozygous condition. And let's go for the homozygous recessive condition. Okay. So what this concept is telling us is that the heterozygote, which is normally carried by the autosomal genes, not the sex genes, the heterozygote will have different effects based on whether it finds itself in a male environment or a female environment, right? So what we're seeing here is that the phenotype here will be the same as the homozygous dominant in the male, but the homozygous recessive will be different. On the other side, what we're saying is this, that the trait, because it's two big A's, the traits will complement each other and produce a characteristic that's the same as the male in the female. But the heterozygote will be different, just like the homozygous recessive. So you have this division here. So in the male, it looks like the homozygous dominant. In the female, it looks like the homozygous recessive. And the reason for this is that there's interaction between the sex hormones that are produced by the two different sexes of individuals in this case. So that is called a sex influence gene. On the next slide, we have a related term called sex limited. And this one could be on the sex chromosomes, it could be on the autosomes. But it's a gene that's limited to one of the two biological sexes, right? So maybe the male has it and the female doesn't, or it could be the other way around. Maybe the female has it and the male doesn't, right? So let's example of where the female has it and the male doesn't. So ovaries. Yes, females have ovaries and males don't. And the other way around will be testes. Males have testes, females don't. And we can keep going. Breasts, right? Mammary glands. Females have them and males don't. So you can see what's going on here. The allele, or the genes, are only expressed in one sex and not the other. So that's sex limited. It's limited to one sex or the other. And the example that most textbooks give is the bird plumage. So male birds have a lot of plumage that they use for behavioral attraction of females. And the female is very drab in comparison. Okay, so we find that quite common in birds, but it's found in many other systems too. So that's what sex limited means. The next one is lethal genes. So a lethal gene is a gene, when present in its lethal allele combination, will kill the individual. It may kill it early on during development. It may kill the individual in mid-age, or it may kill the individual in late age. But we do know this, that 1/3 of all the genes in the human being that we know about are lethal. These genes are normally involved in essential tasks inside a cell, such as replicating DNA, translating proteins, or most commonly regulating the cell cycle so that the cell can divide and produce new cells as the fetus and the embryo develop. Let us focus on a classic example once again. And this time, everybody likes to share the example of a certain type of cat called the Manx. So these cats have a defect produced by the mutant allele, which is dominant in this case, that causes a developmental issue with the spine. So the spine doesn't develop properly and divide, therefore the tail is truncated. So if we take two Manx cats, and we'll use big M to represent the mutant allele and small m to represent the normal allele, and we breed them together, then we'll see that according to Mendel's laws of segregation, we should get a 3-to-1 ratio in the offspring of this particular cross. So we have to put down the alleles outside the Punnett square, and we'll see that this parent produces these alleles and this parent produces these alleles. So we have a typical 2 by 2 Punnett square. There we go. And what we're going to do now is put in the genotypes. So here we have big M, big M. Here we have big M, small m, and big M, small m, and small m, small m. So as I told you earlier, the small m allele makes the cat have a normal tail. So this offspring here will have a normal tail. This condition, the heterozygous, will give it the Manx phenotype. Likewise, this box here will give it the Manx phenotype. This condition here, the two alleles are both damaged, therefore the embryo cannot develop any kind of spine, therefore it is lethal. And these cats are never born. So when something is not possible, we remove that as an option for the children. That makes sense. If you're never going to see that type of child, take it away. So this becomes a 3-box Punnett square. And therefore, you have to then call it as you see it. So the genotypic ratio, in this case, is going to be 2 Manx cats to every 1 normal. And in terms of the phenotype, that also is going to be 2 Manx cats to every 1. So we're not seeing the 3-to-1 ratio, we're seeing the 2-to-1. And that pattern suggests that we have lethal genes inside that system. Just like before when we were talking about the environment, some genes are sensitive to the environment and they're also lethal. So we call those conditional lethal alleles, right? The allele of the gene. The classic example, again, is temperature-sensitive lethal genes. So these are genes that in fungi, yeast, at 1 temperature, the yeast grows well. And it grows just like the wild-type variety. But in a different temperature, the yeast doesn't do well. And it either doesn't grow as well or it dies. In this case, at that higher temperature, maybe 41 degrees, the yeast will be dead, and at 37 degrees it'll be doing quite well compared to the wild type, which will be doing well at both temperatures. So here we have a condition that's lethal to these particular yeast cells. So temperature sensitive lethal alleles. And then finally we have something called semi-lethal alleles. As you would be aware, semi-lethal, I'm half dead, right? No, you can't be half dead. You're either dead or you're alive, just like being pregnant, right? So semi-lethal alleles are an application to a population. So under some environmental conditions, some individuals in the population will be deceased. Other individuals in the population will still be alive. So that can have a percentage. So you can say 30% semi-lethal alleles, right? And that again goes with the fact that the genes are encoding something and the environment is then intercepting that information and then deciding whether it goes forward to its destination or it gets intercepted. So this is like our truck carrying the cargo from the ribosome to its destination. Under certain environmental conditions it doesn't make it and it's lethal, and in other environmental conditions in the same population it delivers its cargo. Okay? Or maybe in the same environmental conditions, different people have different susceptibilities. So let's move on next to pleiotropy. Pleiotropy. Pleo means many, tropy means effects. So a single gene with alternative alleles could produce a plethora of developmental or physiological, anatomical effects on the body of that individual. So the gene is not restricted to just one outcome. Right? Again, most books tend to use the same example, and we can use the same example as them, cystic fibrosis. So cystic fibrosis is a recessive disease in which people have a hard time producing mucus of the right consistency. So what the gene does, the mutant gene, because it's a recessive gene, right? The small a produces a defective protein in the membrane of cells of these patients. So it's a chloride ion transporter. It transports chloride ions from inside the cell to the outside, and then that drags water with it, and that makes the mucus that lines the surface of these cells. So you can imagine any part of the body of this particular patient that requires mucus could be affected. So a single gene can affect many parts of the body in different ways. So different ways is where you get your pleiotropic effects. So in the lungs, it can cause bacterial growth. In the digestive system, it can cause problems with fat absorption and protein absorption. So you get malnutrition, as well as the difficulty breathing, right? In other parts of the body, the reproductive structures, where the mucus is necessary for protecting against infections. Maybe this person has UTIs a lot, and the reproductive system infections, right? So you can see a single gene can have many effects. What we do know now, from all the research that we've done, the examples of the genes that we gave you earlier today are the exceptions. Most genes work at this level, where they interact with many other genes simultaneously to produce the outcome that you have. Remember, we have about 22,500 genes. We don't know the number because we're still finding genes. So those 22,500 genes have evolved together. They've grown up together over eons of time. And therefore, they have complicated relationships. Just like you have complicated relationships with all your relatives and friends, these genes have been around longer than you, and they have even more complicated relationships. So it's not surprising that genes, when we look deep enough, they tend to have pleiotropic outcomes. Let's now move on to epistasis, one of my favorites. Here we're going to be looking at two different genes, and they can be anywhere on the chromosomes, right? And I want to bring back my diagram because now it will make sense to understand this. So let's assume that we have a single gene. Let's put it together so we can get the color right. Here we go. So let's use green this time. So we have a single gene over here. And this gene comes in homozygous, it comes in heterozygous, and it also comes in homozygous recessive. And this gene is responsible for running the conveyor belts in our factory, right? So it's producing product. That's what the gene does. So it runs this. Then we have another gene, and let's call it red. And that gene is responsible for running the truck. Remember, the truck takes the product from the factory to the destination, which happens to be your house in this example. And that gene can also exist in various forms. So we're going to keep it very simple. We're going to keep it this dominant recessive relationship. All right, so let's see how this works. So if you're normal, let's say big A is normal, everything's working fine, and big B, the truck's working fine, right? So if you're this condition and this condition, your factory's working normally. So let's put factory is working normally. And then your transport system is working normally. And then the delivery will arrive, right? The factory produces it, the truck delivers it. So everything is okay. So gene A and gene B are both working together. In this particular case, gene A is running the factory and gene B is running the truck. And let's assume that in this heterozygous condition, the cell is still happy, right? It's a typical dominant recessive relationship. So there's enough product made to satisfy the cell. So the factory is running, okay, and the delivery trucks are running, okay, and the destination is delivered to. Great. Now, what happens if you have this particular combination? The factory is broken, so it's not going to produce anything, and then the truck has nothing to deliver, and therefore nothing will be delivered. Now let's look at an example of epistasis in action. Suppose we have this condition. So let's put it down in a different color because it's so important. Suppose the genotype of the individual is big A, small b, small b. So what would happen in this case? Remember, we're looking at whether you receive the parcel or you don't, right? So the factory is working normally. So we get a check mark here. But the delivery trucks are broken. So even though the product is being made by the nucleus and the ribosomes, it's not being carried from the ribosomes to the destination in the cell where it's needed. And as far as the cell is concerned, if it doesn't arrive, it's not there. So in this case, we get an X right there. The trucks are not working. Therefore, as far as you're concerned, your parcel doesn't arrive. And then you have a detrimental phenotype. So how can you tell the difference between this line here, where the factory is broken, and this one here, where the trucks are broken? Well, you can't. You'll have the same phenotype. So this is an example where a second gene can hide the effects of a first gene. That is epistasis. So let's look at a final example. Say you were this particular configuration. A, small a, and big B, and we could put anything here. Let's put big B. So in this case, the factory is broken, the trucks are working, but there's nothing for the trucks to deliver. So you get the same phenotype as this person over here, even though you have a completely different genotype. So one gene can mask the genotype of another. I think we need to show you an example, a genetic example, and then correlate it with what you knew before and what you will know now. Let's imagine the same scenario where we have gene A producing something inside the nucleus and gene B delivering the cargo. What we learned in the past was that in Mendelian genetics, if we have a heterozygote, a double heterozygote, like this, and we self-cross it or breed it with a like this, we will end up with two systems that we assume are independent, and they will produce a Punnett square. And I won't go into all this, but you will get a Punnett square. Oops, I don't have that. You get a Punnett square that will have 4 rows and 4 columns. And what we have are the gametes from this individual. We have big A, small a. Oh, sorry, big A, big B. I'm carried away. Big A, small b. Now, then you have small a, big B, and then you have small a, small b. Okay, so these are the gametes coming from this individual in all the combinations. Because this individual is the same, we'll have exactly the same as gametes from that individual. Right? So we put them in this very ordered orientation, like this. We'll understand what happens next. What we find in terms of phenotype is that these boxes, and these boxes, and these two boxes, so you get this little triangle here, all these 9 boxes will have the dominant phenotype for both A and B. Then 3 of the boxes, this one here, and this one here, and this one here, will have the dominant allele with homozygous recessive for B, right? So it'll have one dominant and one recessive trait. This one, these boxes here, will have the other reciprocal orientation, where A is homozygous recessive, and at least one of the B's is dominant. And then this final box here, that box will be small a, small b, a double homozygous recessive, and that would be 1. And that gives us back our classic 2-gene Mendelian ratio of 9:3 to 3:1. So that suggests there's no epistasis, right? There's nothing going on, everything is behaving normally. So now let's compare that with a situation in which we have epistasis. So we're going to use the example of the factory because we put that together, right? But let's change the color so we don't confuse the two. And this time we're going to have, let's say F for factory, where big F is the factory is working normally, and small f is where a factory has a broken conveyor belt, right? So in this one, in the big F, the conveyor belt is working normally, and in small f, the conveyor belt is broken, right? And then let's pick a T, the symbol T to represent the truck. So when you have one big T, the truck, at least some trucks are working, and when you have a small t, the truck is broken, right? So let's put a heterozygous together and do the same thing. So we're going to do exactly the same thing over there. We're going to mate a double heterozygote together, like this. And we're going to mate that with another heterozygote, who's this, big T, small t, right? There you go. And now the gametes will be big F, big T, from over here, and then big F, small t, from over there, and then small f, big T, again from over there, and then with one more possibility left over here, that would be small f, small t. All right, and then likewise, because this is exactly the same as this, everything from this will be very similar to that, so we can put it up and down the side, right? So we put those gametes right here. So remember, we're looking for a 9 to 3 to 3 to 1 ratio in this particular case, right? So here we are. Okay. So let's put this across like this, and we'll put the columns in place, and then the rows. All right, so let's start putting things in, right? Big F, big T, big T, big F, big T. Second. All right, FF, TT. All right. So remember, all right, this is the factory. And this is the truck. And we're trying to see if both systems are working together to give us a delivery, right? So we're looking for delivery or no delivery, right? So let's put that there. So the factory's working and the truck's working. So we have delivery. We have delivery. The factory's working and one of the trucks is working, we get delivery. The factory working, the trucks are working, delivery. Factory's working, at least one conveyor belt, and one of the trucks is working, we get delivery. All right, so far so good. Let's have a look at this one. We get delivery because the factories are working, one of the trucks is working. Let's go for this one. Factory's working, trucks are working, delivery. Factory's working, trucks are working, delivery. All right, so it's following this pattern right now, right? Is it not? Good. So let's go and look at this one and this one, okay? So here we are, factory's working, trucks are delivering, delivery. All right, factory's working, trucks are delivering, delivery. So we have our basic pattern, and it looks like it's going to be following the 9 to 3 to 1 ratio, right? So these, this, and these are all going to give us a normal system, right? So 9 means everything's normal, the factory's working, and the trucks are working, right? We're happy with that. Well, let's carry on. We haven't finished our analysis. So let's change colors, and let's use orange, and look at this situation. Here the factories are working, but the trucks are broken, both trucks. Therefore, there's going to be no delivery. No delivery. Factory's working, but trucks are not working. We don't get a delivery. Here, the factory's working, the trucks are not working. We don't get a delivery. Here, the factory's working, the trucks are not working. We don't get a delivery. Here, the factory's not working, but the trucks are working. But what are the trucks going to deliver if the factory produces nothing? So we get no delivery. Here, there's no factory, and there's one truck. Again, there's nothing for the truck to deliver. We get no delivery. Factory's not working, trucks are working, nothing for the trucks to deliver. Factory's not working, trucks are not working, nothing for it to deliver. So in terms of phenotype, we have 9 of the 16 boxes that are saying everything's fine. And then we have 4, 5, 6, 7 boxes that are saying, sorry, no delivery. So now you see a departure from the 9 to 3 to 3 to 1 ratio. You get this 9 to 7 ratio that indicates epistasis, where one gene is influencing another gene. Let's move on to the penultimate slide, complementation. So when you get a compliment from somebody, they are sort of saying something positive for you. But in genetics, complementation means that one gene is helping another. So how do we explain this? We're going to use our delivery example once again. But we're going to have to modify our delivery example to be slightly different. Okay? So what do we do here? Let's consider that one gene, let's use the same example that we used earlier. So let's go with blue. Let's say the T gene, present in two versions. Big T is where this truck is working normally, right? And small t is where this truck will be broken. So big T and small t are referring to this delivery system. Now suppose we have a second delivery system. Yes, we have a second delivery system. The second delivery system is UPS. So we'll use the letter U to represent UPS, and the top one is Truckers United or whatever, right? I just made it up. So the big U stands for a normal delivery truck that's working, and a small u, put it underlined because it looks like the big U, that stands for a broken truck, right? So let's have a look at this. Let's have a look at complementation. Let's assume that the factory is producing green rods that are carried by the green trucks, and the factory is producing blue rods that can only be carried by the red trucks in this case, right? So green trucks carry the green rods and the red trucks carry the blue rods. And you are expecting both types of rods because you're constructing something. So you can't construct something if you only have one type of rod and not the other, right? So you need both to be useful for you. So let's look at some genotypes and see how complementation can be explained. Because we have two genes and we're diploid, we have to deal with 4 letters. So let's put a scenario over here, and let's use a different color for that scenario so we don't get confused. Let's choose blue, dark blue. All right, here's the first scenario. If your genotype is this, big T, small u, small u. If that's you, can your cells deliver for you everything you need to make your structure? Well, this suggests that this truck is normal. And this suggests that this truck is not normal. So you may get delivery of the blue rods, but you don't get delivery of the green rods. Excuse me. And as far as your project is concerned, it comes to a standstill. So this leads to a mutant lack of ability to produce wicker baskets, right? Let's now focus on a second individual, the reciprocal, right? So this person has small t, so they're homozygous recessive, and they have big U, homozygous dominant. So in this person, their red trucks are not working, so they're not going to be able to deliver blue rods to your house, but the green trucks are working, and they are going to be able to deliver green rods to your house. But as far as your project is concerned, it's at a standstill. You cannot make your structure, your baskets, unless you have both colors. So in both cases, the phenotype is mutant. You're not making baskets. And over here, you're not making baskets, right? Mutant, mutant. Now let's suppose we cross them together. We take these individuals and we cross them together. So we're going to do that now. And what's going to happen is we're going to look at their offspring, right? So the gametes from this side, let's get rid of the M because it's in the way, right? So let's get rid of this because it's in the way. Right, so let's go back to the pen. So the gametes from this individual are going to be big T, small u. Remember, the gametes can carry only one of each letter, one T and one U. And there's no other choice, right? And the gametes from this individual here are going to be small t, one of the two T's, and one of the two U's is going to be a big U. And when they fertilize each other, the baby will be born down here. And the baby will have big T, small t, big T and small t come together, and big U and small u, like that. So now the baby is a double heterozygote. That's the genotype. Let's go and look at what's happening at the molecular level. So we have one big T. That means one of the red trucks is working out of the two. So you will get some delivery of blue rods. And one of the two trucks is working of the green type, and you will get green rods. So now you're getting both types of rods delivered to your house, and you can get on with your project, right? And you can make wicker baskets. So this individual was a mutant, this individual was a mutant, but when they bred together, they produced a wild type. And we say that the two genes, the two truck companies in the heterozygous configuration have complemented each other. So they've helped each other to deliver some cargo to your house. That is complementation. Let's finish off this lecture with looking at redundant genes. So the word redundant means extra, right? So when you have redundancy, you have a backup, you have an alternative means of doing something. So extra genes would come about by various mutant methods that we'll discuss in the future. But suppose a normal individual has just one copy of a gene that codes for an important enzyme. That gene is necessary, and it's going to have to be functional, at least in the heterozygous condition. Suppose that gene was copied accidentally into two copies. Now we have the original one doing the same job of maintaining the life of the cell, and the second one is free. It's like a spare. This reminds me of, Prince William and Prince Harry. Prince William is the king, and he'll take over from his father in the future, if his father becomes king. But Harry is the spare, and he can play around. It doesn't really matter. He only becomes important if something happens to William. So as long as the original gene is working, the second gene is free to accumulate mutations without influencing the phenotype of the cell in which it resides. So this gene can accumulate mutations without having to worry about what happens to the life of the cell. So the extra copy can now mutate at a faster rate than it would otherwise, and it may lead to a better function than the original. Maybe Prince Harry may be a better king than Prince William. And if he is, then he can take over at that point. So extra copies of genes are fodder for evolution, accelerated evolution. Now, if the genes are in the same individual and they come through this method of duplication, we call them paralogs. That's just a name for genes that are duplicated in that particular lineage. So redundant genes are good. They speed up evolution, and they become a backup should something happen to your primary gene in the case of mutations. I'm sorry about this huge lecture, but what you should do is watch the parts that you don't comprehend and then speed across or even skip the parts that do. Thank you so much, and good luck.